I'm Honghui Zhang from the Art Institute. Today, we'll present to you the work that has been included in a recent publication package in Nature. The work was from the Brain Initiative. In the consortium, we have extended our cell atlasing work in the adult mammalian brain to creating comprehensive cell atlases in the developing brain from mouse to human. And this new set of effort, we believe, will create a new frontier in better understanding the development of cell types in the mammalian brain and its implications in brain function and diseases, in particular neurodevelopmental diseases. So I'm giving you an overview here on behalf of my co-leads of the package, Arpana Barduri from UCLA and Tomas Nowakowski from UCSF. Over the past decade, Brain Initiative Cell Census Consortia have been using single-cell genomics technologies, as well as other multi-model approaches to create comprehensive and high-resolution cell type atlases in the mammalian brain, from mouse to non-human primate to human. And this work has culminated in several major publications, especially from the second phase of the Brain Initiative effort called Brain Initiative Cell Census Network, or BICCN. And in this publication packages, we have generated cell type taxonomies and atlases, starting with individual brain regions to the entire brain. In 2021, we published a set of 16 papers in Nature describing the consensus cell type taxonomy of primary motor cortex from mouse to non-human primate to human, and reveal a series of underlying organizing principles of the brain. After that, just 2 years later, the consortium also generated a large set of studies about the human brain, and about 20 papers, and one of the central themes of that set of studies is the creation of a draft version of the whole human brain atlas. And immediately following that, again, the consortium and with multiple teams working together, 10 publications in Nature, created a multi-model single-cell transcriptomic epigenomic and spatial transcriptomic atlas of the entire mouse brain with more than 5,300 cell types or clusters identified. So following up on this comprehensive work, now in the third phase of the Brain Initiative called Brain Initiative Cell Atlas Network, or BICAN, one of the main goals now for us is to create similarly high resolution and comprehensive cell atlases across the entire pre- and postnatal development of the brain, again, across multiple mammalian species, from mouse to marmoset, macaque, and to the human brain itself. The goal of this is really to understand the similarities and differences of cell type development across species, understand how the human brain develops in ways that are similar to the animals, and also in ways that are unique to the human itself. And we want to use gene expression changes to correlate with the various structural and functional developmental events, and from there gain mechanistic understanding of how brain development occurs and how it could go awry in diseased conditions. And here summarizes the main approaches that we have taken to create those cell atlases. They include single-cell RNA-seq, single-nucleus multiome, which is the combination of RNA-seq and ATAC-seq, and spatial transcriptomic technologies, and couple that with lineage tracing. And from this kind of a large-scale comprehensive studies, we were able to create cell type classification schemes and achieve cross-species alignment, and reveal the spatial localization and migration of different types of cells during development. And from this kind of atlasing effort, descriptive effort, we also have gained a certain insight into the molecular mechanisms of brain development, including the generation of cell type developmental trajectories or lineages, as well as the gene regulatory networks that may be regulating or modulating the brain development. Finally, we think this kind of work, cell atlasing work, will have a big impact in understanding both the mechanisms of brain development, as well as in discovering ways of better understanding diseases and the treatment of diseases. This includes going from molecular mechanisms to understanding how connectivity and function emerges in the brain, and to the understanding of the different genetic variations or mutations that could lead to selective vulnerability of different cell types in different kinds of neurodevelopmental disorders, identifying specific pathways that may be particularly changed diseases. And then for better study of disease models in both animals and in vitro systems, and helping with the functional evaluation of potential drugs or genetic interventions, and ultimately may lead to the development of precision medicine. Of course, this is a long way to go, but we think that having comprehensive high-resolution cell atlases would be essential, would be serving as essential foundational resources to enable all these things to happen. Okay, so with that, I'll end my introduction, and you'll hear individual talks now about the insights that the team has gained from a set of really spectacular studies, and I hope you will enjoy the talk. Thank you for your attention.
Thank you, Hongri, for that introduction. Next up, we have Carlo Colantuoni, who is adjunct faculty at Johns Hopkins and the Institute for Genome Sciences of University of Maryland.
Thanks. I'm very excited to share our work today in this incredible effort that Hongri just gave us an overview of. Our study is quite different than most of the others you'll hear today in that we generated no new data. So our entire mission was to get all of the data we possibly could together in one place, not only to enable our own research, but to empower researchers across the globe. And we focus particularly on neocortical development. And we really want to get this in the hands not only of bioinformaticians, but as I'll show you, everything I show you today can be point and click online at NeMO Analytics. Okay. So the first piece of what I want to show you today is the data, a little bit about it, and then what we can do with it. So this is kind of a summary of what's in NeMO Analytics. We have data from mouse, primates, and human, just about as much as we could find and bring together. And that's both in vivo development and also in vitro models, such as iPS cell-driven neural differentiation and cerebral organoids. So that's where you can go online to surf data sets looking one gene at a time. What I'll talk mostly about today is how we can grab focused subsets of these data sets and understand them in a deeper context together. And one thing that we'll highlight is the other thing that you can do online, in addition to looking individual genes, is to explore complex gene signatures from your own analysis or from across the literature, across this same expanse of data that we've brought together. So here's our first example of an analysis. We do a lot of joint decomposition. Please reach out if you're more interested in the methods. I won't talk about them today. So here is in 3 rows, 3 data sets: mouse, monkey, and human, single cell RNA sequencing from mid-gestation, focusing on the excitatory neuronal trajectory. So looking at the birth of glutamatergic neurons in the neocortex. What the 4 columns are to the right are 4 different signals that we got out of the joint decomposition that are found to be common across these systems. So this is sort of a high resolution version of what we know to be true. That's why we call it the neocortex. It's new in evolution. It's in mammals, and we know the 6 layers are conserved, and we know some of these states are conserved. So these are 4 patterns that we got that are shown to be conserved across these 3 data sets. They correspond to radioglia, IPCs, intermediate progenitors that come from those, and then new neurons that are being born, and finally in the last column, maturing neurons. So here we're looking at the sample space, so the cells in each case, in each data set, in each species that are expressing these different patterns. But behind this is the gene space that describes which genes across the genome are being harnessed in each of these stages. And now we're going to take those signatures. So if I did this offline, I can upload those signatures to NeMO Analytics, and we can explore them in further data to get a much deeper understanding of where those signals came from. So the 4 columns I'm going to show here are the same 4, the radioglial signature, IPC signature, new and maturing neurons. And as I go along, I'll show you a new data set, just where these signatures are showing up. So this is bulk RNA sequencing from laser microdissected human cortex. So in the first column, you can see the radioglial cells are showing up in these early ages in the ventricular zone, right where we know the radioglial cells to be. The next column, where the IPCs have delaminated from that ventricular surface, that molecular signature is now showing up in the inner subventricular zone. And then as the neurons are born, they're still in those subventricular zones. But by the time a neuron is expressing this last signature, it is in its final resting place in the laminar organization of the cortex in the cortical plate. So this is a much newer spatial study that can give us even higher resolution understanding of where these patterns come from. This is StereoSeq. And again, we're looking at each of these same 4 patterns. You can see that the radioglial signature lines the ventricle here. The IPCs move out and so on. And really, it's just the exquisite data that can be produced today enables this kind of analysis. So, and again, you can see additional stuff you weren't even thinking about asking, perhaps. We were looking in the telecephalon for these signatures, but the progenitors and also neurons of the entire brain express these signatures in different times and places. And timing is key here. This is now looking at these same signatures in a study where they birth dated every cell before they sequenced it. So you know the last time this cell divided before you get all its transcriptional information. So this allows us, at least in the mouse here in this experiment, to look at how long it takes a cell to move through these states. And this now is the same 2 signatures, the nascent and maturing neurons, showing that they turn on the nascent signature and then turn off the maturing one. So if you kind of keep this in mind, we're going to connect this after one slide to the end of the talk. So quickly, I'm going to reverse the sort of direction of analysis here and then come back to what we just looked at. So we took some adult data, even though we're interested in development, and got a lot of single nucleus data from the adult neocortex from the Allen, and we decomposed it into 20 patterns. These rows are 9 of those patterns, where we can see across spatial and single cell data that without any instruction or supervision, the algorithm can identify very layer-specific signals. These are 2 of those, a layer 4 signature on bottom and a layer 3/4 signature on top. And I just zoomed in here to show you the wonderful data that's out there for us to use. So you can see in these 2 different human studies where exactly, and there's really sort of fine details right in layer 4 or just above it. This is in the macaque, another wonderful StereoSeq data set. And when you can look at so much data so sort of smoothly and in a directed fashion, things start to fall out. For instance, in this mouse data set, we discovered that this layer 4 signature does not occur in the mouse cortex, just because we could see it. So now we started with those developmental signatures. We now have a collection of adult signatures. This is now starting to bring those together. So this is that layer 4 signature that was primate specific. We're now in the human, so it's showing up. The color scale here shows you age, where in a specific layer 4 population, this is coming on, but it's not just coming on during in utero or neonatal development. it takes many, many years, and it looks like about a decade for this particular signature to reach its maximum. Now, this is interestingly in stark contrast to canonical transcription factors that are known to set up these patterns. So RORB is a layer 4 master TF, but it's peaking right about at birth and then trickles down. And I think Aparna Baduri, who will speak today, will also give you sort of a in-depth view of one gene, FESF2, that plays in a specific game of this general lesson. Okay, so last thing I want to show you is, again, bringing all this together. So this is a data set that has both in utero and postnatal samples. We're looking at that turning on of the nascent signature in neurons and then turning on maturing signatures as that goes off. That's what the up and down is here. And the color is the color of one of those, or the strength of any one of those 9 layer signatures. So if we look at one row, this is when and where the layer 3-4 signature comes on, right about here at 22. So if we extend this, all of a sudden we can look at every age as cells go up and down in this process, where do they turn, when and where do they turn on the adult signatures? And these green arrows can kind of show you that just like the inside-out organization of the birth of neurons in the neocortex, the maturation follows the same trajectory, with a notable exception of one of these patterns that shows up 10 weeks early, which is super interesting, and we're looking more into that. The last thing I want to show you is this same analysis now, but looking at cerebral organoids. We need to know what organoids model so we know how we can use them and how we can improve them. So we're going to do the same analysis that we just did in vivo development, but now in organoids. So the 9 rows are, again, those signatures from the cortex of layer-specific neurons, and the columns are time points. And really, there's only 2, in this and in many other data sets, there's really only 2 deep layer neuron signatures that show up to mature well. Now, I'll show you one last thing here. One thing that you can push you much farther, this is from Sergiu Pasca's lab. If you put that organoid into a newborn mouse pup's brain, all of a sudden everybody is much more happy to keep maturing, with the notable exception of an upper layer signature, and that layer 4 signature that we saw was absent from the mouse does not get induced, even though these are human cells, the mouse brain environment can't hit it. So I'd stop by saying, please get your own data in here if you have new data, and please reach out and use these resources in your own research, whether you're coding or clicking. Thank you very much.
Great. Thank you, Carlo. Next up, we have Aparna Baduri, an assistant professor of biological chemistry from UCLA, and she'll be presenting on behalf of a postdoc in her lab, Patricia Nano.
Hi, everyone. Hopefully the sharing is working well. I am really delighted to see all these papers out, to see the package out, and I'm delighted to present today some of the work from our paper in the package, which is led by first author Patricia Nano. Unfortunately, she couldn't give this talk today because she is at a faculty interview. I think she might be literally mid-chalk talk, so sending good vibes her way and excited to share what she's been working on. So the study that we presented as part of the developmental working group package was a meta-atlas of the developing human cortex, and the goal was to, similar to what Carlo was doing, but with a different approach, identify modules that are driving cell subtype specification in the developing human brain. So the rationale for doing this is basically that there's a lot of molecular profiles that have been made. At the time of our study, this was primarily from some of the initiatives that Honghui had highlighted as being previously published, and this included some of the previous Brain Initiative packages. However, not all of these studies can span all of human brain development, and this is really a function of some of the rare samples, as well as some of the cost considerations and the availability of human tissue, which is going to have many technical differences across different modalities, as well as a function of the fact that it is indeed human tissue. So to this end, Patricia wanted to generate a meta-atlas that would put together these data sets and allow us to find broader biological patterns that might be describing some of the trajectories of development. To do so, she leveraged 7 single-cell RNA sequencing datasets that focused on the peak stages of neurogenesis. This came from 96 individuals, and after our additional QC to have uniform standards across the data, it was approximately 600,000 cells. And to begin for visualization purposes, we used a standard RPCA type integration, but we wanted to be able to look not only at the cell types and clusters that were emerging from traditional analyses, but also to overcome some of the technical variations that are going to vary based on things like sequencing depth and single cell capture methods, as well as to identify subtle differences between developmental cell types. And the overall goal of this study was really to start to begin to understand how we can link gene networks in cell biology. So to do this, Patricia innovated a novel gene co-expression analysis method, which we are calling a meta-atlasing strategy to make meta-modules. The way that this works is it looks at each of the 96 individuals in the meta-atlas one at a time. This allows us to use unsupervised clustering approaches for each individual in order to identify the axes of variation that are present in that individual sample. This helps us overcome some of the technical variation, and when we then look at the gene expression signatures within each individual and compare them between individuals, we can start to get these meta-modules, which are networks of genes that are changing across different samples in the data set, but also that are important for any given time point or sample. With this approach, Patricia generated 225 meta-modules, and she annotated these using a combination of prior knowledge, literature, gene ontology enrichment, etc., and was able to see that there was a mix of modules. Some of them seemed to be cell type specific, indicating that there were cell types represented that maybe are specialized in something like axon function, whereas others seemed to be broader biological processes, such as those related to metabolism, that we thought might be more continuous across the spectrum. In order to look at this more deeply, we wanted to make sure that this was in fact the case, so we used some positive controls, such as looking at vascular modules in vascular cells and microglia and immune modules in the relevant cell types. We also observed some specificity of glial populations and those relevant modules, but these were sparse given this focus of the peak stages of neurogenesis. Now, one of the questions that we had was, should we be doing this from a developmental forward-looking approach, or should we be looking backwards from the adult cortex into development? And we wanted to be able to ask the question of which of these approaches is going to allow us to understand the initiation, refinement, and mechanisms of cell type specification. So to answer this question, Patricia took her meta-module approach for a spin and looked at an adult meta-atlas that she also generated. This was primarily derived from some of the papers that were published as part of the Brain Initiative Adult Atlas that came out in I believe 2022 or 2023. And from this was able to compile 16 data sets across 274 individuals and 2.6 million cells using the same approach. This now gave us an opportunity to compare modules between the two contexts. And one of the ways that Patricia did this was using a module specificity score. She did this by looking at the specificity of a given module, which are shown at the top of this heat map, across cell types, which are shown in the rows. So here you can see what the developmental modules look like when applied to development, and you can see a lot of that continuity and co-expression that we were discussing and has been well known to exist in development. Similarly, when you look at adult modules on the adult brain, you see something very different. Patricia likes to use the word crispy to describe this, and I kind of agree with it. It really shows a lot more specificity to individual cell subtypes or groups of cell types. So Patricia then asked, what happens if you apply the developmental modules onto the adult or the adult modules onto development? And what we observed is, regardless of the source of the modules, there was always more continuity in development and more granularity in the adult, which suggested to us that if we want to understand how these cell types are made, we need to focus on how that specificity comes from the broad expression that is seen in development. So focusing on development first and then looking forward into adults might be a good strategy to identify mechanistic modules. I'll give you a quick vignette on how we did this for one of the modules. Module 20 was a module from the developmental atlas that was expressed in neurons and seemed to increase over time in deep layer populations, but not in upper layer populations. This was particularly interesting because when we looked at it in the context of the adult brain, we saw that it actually did have some specificity, as was alluded to. And in this case, it was specific for FESF2 annotated subtypes. Now, this was really interesting to us because FESF2 is one of the terminal transcription factors that has been described in the mammalian brain, and a lot of beautiful work has been done in the mouse to describe how FESF2 is capable of specifying specific subtypes of deep layer neurons in the brain with very unique projection patterns. Now, what was interesting is that FESF2 was not actually a member of the Module 20, but its expression preceded that of Module 20's peak. And when we looked at some of the existing mouse data sets that exist that looked at ChIP-seq to look at targets of FESF2, we saw that more than half of Module 20 genes were in fact FESF2 targets, suggesting that this might be a regulatory mechanism. We also saw that the main transcription factor, TSHC3, was in fact one of the regulated genes, and this was the only transcription factor that existed in Module 20. So we wanted to test whether FESF2 and TSHC3 are required for Module 20 activity, and we did this by doing a knockdown experiment in cortical organoids. I won't go through all of the details, but basically we leveraged a chimeroid system in order to look at multiple lines simultaneously. And after generating these organoids with knockdown at 8 weeks, we sorted out the knockdown cells from FESF2, TSHC3, and the double knockdown. What we saw is that there was a decrease in Module 20 activity, as well as the expected decrease in deep layer populations that seemed to trade off with an increase in radial glia. Now, what was really exciting about this is both FESF2 and TSHC3 seemed to regulate Module 20 genes based on the RNA expression. But when we looked at chromatin accessibility from our multiome profiling, we observed that TSHC3, not FESF2, was required for chromatin accessibility of FESF2, TSHC3, and Module 20 genes, suggesting a new model where TSHC3 is an important intermediate in the specification of deep layer neuron identity, where waves of expression of FESF2, TSHC3, and Module 20 are required for this deep layer neuron specification. If you're interested in digging into this further, the meta-atlas is available at the UCSC cell browser, both for development and adult, and the code to make your own meta-atlas is available on GitHub. With that, I'd like to conclude. I'd like to thank Patricia and all of her colleagues and co-authors on this study, and I will now hand it off to the next speaker. Thank you, everyone, and I'm happy to answer questions in the Q&A chat.
Great, thank you, Aparna. All right, next up we have Di Zhang. She is a postdoctoral fellow in the Department of Biomedical Engineering at Yale.
Great. Thank you for the kind introduction. Good morning, everyone. My name is Di Zhang. I'm from Department of Biomedical Engineering at Yale University. Today, I will talk about how I developed and applied the spatial multi-omics technologies to decipher the mammalian brains, asking how cells coordinate, make decisions, and become dysregulated within their native tissue environment. The brain is the most complex organ in the human body, orchestrating thoughts, emotions, and behaviors through intricate molecular and cellular interactions. As we know, each cell was regulated by proteome, transcriptome, and epigenome, including open chromatin, histone modifications, and other processes. We realized spatial epigenome and transcriptome co-profiling by using deterministic barcoding in tissue, or DBIT. DBIT is a versatile platform. The key idea is that the barcode's information can be written into the tissue section. Beyond that, To investigate the molecular mechanisms across all the layers of the central dogma, we further realized our ambition of doing spatial triomics mapping on the same tissue section. Scientists for brain development research has mainly focused on neurons. However, the non-neuronal cells also require our attention, such as understanding when, where, and how does myelination occur in the brain. Therefore, we mapped the mouse brains from newborn to juvenile stages using spatial triomics, which received clear spatial UMaps for both attack and RNA. The mature oligodendrocytes marker MBP showed RNA attack and protein signals at P10 and P21 in corpus callosum. The gene FOXP4 involved in regulating morphogenesis disappeared during development. Perhaps is no longer required when all brain structures has formed. We further integrated our attack and RNA data, leading to a more refined spatial domains, which provide better arealization when compared to multiplex immunofluorescence imaging. We dive into the corpus callosum and found that the myelin signals start emerging at around P7 to P10 at the lateral parts of the corpus callosum, reaching full coverage by P21 and extending into the cortex, suggesting a lateral to medial progression of myelination. To investigate the spatial and temporal variations, we developed a computational framework to model attack and RNA signals in corpus callosum. After modeling, we observed not only concordance between the attack and RNA signals for oligodendroglial genes, but also chromatin priming at P0 to P5, where RNA expression is observed at later stages. We also applied the same computational framework to the cortical layers. We found alignment of RNA and attack signals for many of the layer-specific transcription factors, such as COX2. However, we also observed misalignment, such as MEF2C. This might reflect an epigenetic memory of the developmental states. After combined analysis of all the genes for projection neurons and myelin in each spatial pattern for both cortical layers and the corpus callosum, we observed a timed and sequential bidirectional myelination process occurring from the central corpus callosum, where the OPC-COP enter from the postnatal wave to first migrate and differentiate in the lateral part of the corpus callosum, and then followed by the second phase of migration and differentiation in the medial direction. We further use a retrograde AAV tracing strategy to trace the preference of myelination and found that while cortical thalamic projection neuron axons in the lateral part of the corpus callosum are already myelinated, the closal projection neurons in the medial direction are starting to be myelinated, indicating that the axon of neurons might orchestrate the OPC migration to MOL myelination process. The different cells are not separate chapters, but as movements of the same symphony of time. Neuroinflammation recycles many of the developmental programs and occurs in almost every brain disorder. In multiple sclerosis, the immune system attacks the myelin sheath around the neurons to cause demyelination. MS is difficult to treat because the exact cause is unknown. We built a focal demyelination mouse model of MS to study the disease process. We found that the myelin and the mature oligodendrocytes were disappeared at the lesion site by 5 days, with macrophages and microglia populated there, marked by CD11b. By 21 days, The lesion was much smaller with clear remyelination and reduced macrophages and microglia. Here we zoom in on microglial-enriched regions in the primary lesion and in distal white matter regions. We found that distal microglia are more neuron supportive, while microglia at the lesion core are pro-inflammatory. Importantly, inflammatory signals in the distal region appear later than in the primary lesion, suggesting a delayed wave of inflammation. We further mapped the sagittal brain sections. Interestingly, we found distal microglial activation spreading in anterior, posterior, and ventral directions, but returned in the white matter tracts. How about the situation in the whole mouse brain instead of just one tissue section? Using 3D trick discal imaging, we saw microglia spreading into both hemispheres with stronger signals on the injection side. These findings suggest that a focal demyelinating lesion can trigger distal microglial activation, potentially propagating inflammatory signals along axon tracks. In summary, we generated the spatial triomic atlases of mouse brain development and demyelination. We found that the layer-specific projection neurons can coordinate axon genesis and myelination during development. In focal demyelination model, we uncovered microglial activation in distal white matter regions, potentially transmit the inflammatory signals through neuronal circuits. Using spatial multi-omics, we are not only observing cells, but we are witnessing how life organizes complexity into order and how it rebuilds itself amid chaos. I would like to thank all the grants and my amazing collaborators for supporting this work. Thank you for your attention.
Great, thank you, Dee. Next up, we have Young-Sung Kim, who is a professor in the Department of Neuroscience and Experimental Therapeutics from Penn State University.
Hey, hello everybody. Today we're gonna talk about the developmental atlas framework called Developmental Mouse Brain Common Coordinate Framework, in short DEV-CCF, as a part of this consortium effort. Let me get started. Through the BICAN effort and many other brain initiative effort and many also individual effort, there has been a huge advancement to understand the brain cell type composition and their connectivity and their molecular profiling and all these informations. And one of the key aspects of this large scale effort is to map all the data in the coherent spatial framework. In an adult brain, for example, I'll let mouse brain common coordinate of version 3, CCF V3, serve as a common coordinate systems to map all these informations, which has been really critical to integrate this multi-scale and multi-omic multimodal data set. In the developing brain, before we started the project, there was a few two-dimension atlases based on histological section. So this is Allen developing mouse brain atlas based on histology section and then the segmentations. Although this provide very detailed segmentation in the based on histology, these types of two-dimension atlas cannot support the 3D atlas, a 3D data set. To fill that gap, we started projects to establish multimodal 3D atlas in the developing brain. And we decided to use two different imaging modality. The first modality is MRI. MRI provide in-skull data set, though relatively low resolution, it provides in-skull resolution in the intact volume. And the brain can be dissected out and then undergo tissue clearing process, and we use a light sheet fluorescent microscope to acquire cellular resolution informations. And then this light sheet data is also mapping back to the MRI-based atlas framework. And we use a combination of different criteria to segment the brain areas across the entire mouse ages. And our chosen time point is between embryonic day 11.5 all the way to the P56 in 7 different stages. So I'll provide a bit more detail how we accomplish these goals. So the first part is developing template. And previously, mouse atlas is based on traditional N=1 brain. We want to change that. So this time, we pulled about 10-ish from the male and female mouse brain at each age to generate morphologically averaged and intensity averaged template across 7 brain stages. Here is MRI template, and we also have a light sheet based template with a lot higher resolution. And because light sheet imaging require tissue clearings, and tissue clearing often include unexpected tissue distortion, we align all our light sheet data back to the MRI space to bring the morphology back to the intact space. And with this, now MRI and the light sheets are in the synchronized space. And because MRI provide 3D coordinate, we were able to put the grid to calculate, to display how this template placed in the 3D coordinate systems. Not only this, we also imported RNA-CCF3 from adult brain into our adult MRI and the light sheet based templates to put the RNA-CCF3 in the stereotaxic coordinate frame as well. With this, we were able to establish template across 7 different stages. After establishing template, we need to establish anatomical segmentations. So the way we draw anatomical segmentations is following. So here is a light sheet autofluorescent signals, and you can see 3 different plane. And here is MRI, ADC contrast, MRI DWI contrast, and FA contrast, and T2 weighted contrast. And these different contrasts provide a different contrast in different brain region that help us to delineate and establish anatomic labels. And I want to emphasize the fact that we use a different anatomical label compared to the adult CCF3 labels based on cytoarchitectures. In the developing brain, cytoarchitecture change rapidly. So we have to use a development-based atlas label scheme. So we borrowed the development ontology-based scheme pioneered by Luis Puelles. And here we were able to establish 3D atlas also across all 7 ages. And when we establish these anatomic labels, we use various different evidence, including contrast from the different imaging modality, as well as antibody label that light up particular features of the brain, and also including initial stainings as well. And with these anatomical labels, we were able to calculate how the volume of the mouse brain increased across the developmental time frame. So what is the use of this? To demonstrate the utility of these newly established anatomical frameworks, we use a transgenic animal that labels GABAergic cell type in the developing brain. We use a GAD2-Cre crossed with a Cre-dependent reporter mouse and perform tissue clearing and map their expressions. So here is the raw data. You can see GABAergic cell types emerge in the different parts of the brain, and this is embryonic day 11.5, 13.5, 15.5. When their signal is detected and maps onto the atlas framework, we can clearly see how GABAergic cell type in different brain areas emerge across the time from the different areas. So those are development time point. And our development atlas also include adult time point. And this adult time point give unique opportunity to compare and contrast our developmental-based atlas labels to side architecture-based CCFV3 labels. What you see on the left is our RLAN CCFV3 labels, and one on the right is our DevCCF-based labels. And because both atlases are in the same template, we can compare left side how DevCCF label correspond to the right side RLAN CCFV3. And leveraging this atlas being in the same space, we were able to import the spatial transcriptome data from Allen Institute published in 2023, and then compare how these different cell taxonomies is located in different anatomical segment. The top is based on an anatomical segmentation in the CCFV3. Bottom is our DevCCF-based arrangement, how these different cell types are arranged. And to highlight the difference between DevCCF and the CCFV3 anatomical segmentation, I want to draw your attention to this particular brain area called P3. And the P3 is prosomia 3 in the DevCCF. And this is a combination of a zona incerta here. And this is considered as a part of a hypothalamus in the RLAN-CCFV3. And here is a reticular thalamus as part of area considered as part of a thalamus. But this combination of both thalamus and the zona incertas are considered as a one structure, same structure called prosomia 3, based on our developmental ontology-based anatomical segmentations. So this provides perspective how anatomical segmentation can differ based on their criteria. So we established all this atlas framework. We wanted to be used and shared as broadly as possible. So we built a neuroglance-based data sharing platform. So if you come to this website, our lab visualizers, you can see DevCCF resource. And here is a download link. All these files, including high-resolution data, are viewable and downloadable openly from these links. And I want to summarize it. So what I show you today is we established developmental atlas with a 3D template based on MRI and the light sheet, and also anatomical labels based on developmental ontology. And we demonstrate use of it by mapping GABAergic cell type in the developing brain. And we also shows how developmental common coordinate framework label is compare and contrast to the site architecture-based CCFV3 labels. With this, I want to acknowledge that Faye Krugman was our brilliant former graduate students, and Faye led the entire project with great collaborators. And Jiang from NYU provide MRI support, and Luis Puelles was pioneer of developmental ontology-based segmentations. And Lydia and Jim provide neuroinformatics support. And I also appreciate all the contributors and the funding source from NIH. Thank you.
Thank you, Young-Soo. Next up, we have Ruan Gao from the Allen Institute, where she is a scientist on the informatics and data science team.
Hi, everyone. I'm Yuan Gao. I'm a scientist at the Allen Institute. So today I'm very excited to share our work, continuous cell type in diversification in mouse visual cortex development, where we explore how the cell types develop and evolve across ages in the mouse visual cortex. So our data set spans from E11.5 to P28, and we also supplemented this data set with the adult P56 visual cortex data from the whole mouse brain project. And this schematic highlights the major milestones in the isocortex development across the ages. We collected the 10x version 3 single-cell RNA-seq data and also the 10x multiome data. After quality control, we retained more than 500,000 cells from the 10x version 3. and over 200,000 nuclei from the Multium dataset. The bottom panel here shows the number of cells that pass the QC at each of the age. This is the overview of our analysis workflow from this study. And after collecting data from each of the individual time points, as I showed in the previous slides, we do the clustering within or between different ages. These steps serve for different purposes, including identifying the major developmental patterns and also the cluster level quality control. And then we do the integrations between ages, and we systematically propagate the cell type labels from the later stages to the earlier stages. This allows us to connect the adult cell types to their developmental origins, and also to reconstruct the trajectories that trace each of the adult cell type back to the developmental counterparts. In the meantime, we use the Murphy data to validate those cell types. So once the trajectory was built, we analyze the gene modules that are co-expressed across different ages and also the cell types. We also examine the gene trains within each of the subclass to understand how those gene functions shift during maturation. In parallel, we also collected the Multium data, and we integrate our single-cell RNA-seq data and the Multium data. Then we transfer the cell type labels from the single-cell RNA-seq data to the Multium data and leverage both the transcriptomic and epigenetic information to build the gene regulatory network. This enables us to identify the key transcription factors that drive the cell type specification and also the maturation. Our study finds very high transcriptional diversity among the early embryonic cell populations, which may indicate that the cell phase specification may occur at very early stages. These three U maps are for the embryonic population colored by subclass, age, and cluster. As you can see the U map, this is a non-IT population, this is IT population. We can trace the divergence of the non-IT and the IT population in the stage of the intermediate progenitors. And even within radioglia, they show dynamic temporal changes in gene expression, as you can see in this dot plot. In this plot, we can see the markers of the non-IT cells, for example, the FASF2, the IT cells like POS3F2, and also markers for the TNC. Those emerge in a staggered overlapping pattern, which becomes increasingly distinct at the later developmental stages. Our analysis also uncovered the early signals that drives the diversification of the OPC and astrocytes. In addition to that, we also found a distinct population, which is glioblast SVZ. This population gives rise to a specific adult astrocyte cell type localized in the subventricular zone. We also constructed the detailed trajectories for the emergence of each of the subclass with the precise time steps. And our findings indicate that almost all the subclasses we observe in the adult stage, they are already specified either before or soon after birth around P0. The only exception is the mature oligodendrocytes, which appear at about P10. And to study how gene expression changes across development, we have identified about 5,000 developmentally regulated genes, and we group them into 36 trajectory patterns. These patterns fall into 5 broad developmental trends, which is up, transient down, and there's one more which is constant at different levels, which is not shown here. Even within the same category, individual patterns can differ in meaningful ways. For example, this is trajectory 2, and this is trajectory 3. The trajectory 2 increases during the early embryonic stages and plateaus at about P0, but trajectory 3 increase mainly after birth and plateaus around the time of eye opening. Most of the genes show similar patterns across related subclasses, but many also show subclass-specific dynamics. For example, as you can see here, this is for TBR1. TBR1 expression broadly increased during neurogenesis, but it diverges across the upper and the deep layer neurons after the immature neuron stage, with the strongest downregulation occurring in the layer 5 ET cell types. And several other key transcription factors, for example, COX2 and FOXP2, they show similar complex and subclass-specific temporal regulation, highlighting their tightly controlled roles during the cortical development. In the postnatal period, we examined the transcriptomic changes across the subclasses. The plot on the left shows the magnitude of the gene expression changes between the consecutive time points, where the positive values indicate the upregulation and the negative values indicate the downregulation. This is for the non-IT cells, and this is for the IT cells. In this plot, we observe multiple waves of changes, and over time, with the eye opening around P10 to P13 marks the final bigger wave of change. This is not only observed in the non-IT and IT cells, we actually observed in most of the cell types, including the GABAergic cells and other non-neuronal populations, which is not shown here, and we have more details in the manuscript. Activity-dependent immediate early genes show the strongest responses. For example, NRN1, which is triggered by the calcium influx, shows dramatic upregulation, and other well-known classic IEGs like BDNF, NR4A1, EGR3, they all increase after eye opening, although with different cell type-specific timing. Using the gene regulatory network analysis, we identified that the AP1 complex is the central regulator of the activity-dependent maturation. AP1 and BAC2, they share many target genes, and our analysis suggests that BAC2 is downregulated after eye opening, and it normally represses the late onset genes. Its downregulation opens those sites, allowing the AP1 such as Jun and FOS, to activate those target genes and promote the neuronal maturation. We also identified the cell type specific regulator. For example, NR4A2, as you can see here in the network, is a key regulator of the CAR3 subclass, with its motif enriched in both early and late CAR3 subclass specific PIP modules. And also in the non-IT cells, the PO3F1 is a regulator of the L5ET, along with their cell type specific targets. Finally, I wanted to thank all our authors who have contributed to this work. Especially, I wanted to thank Honghui, Zijian, and Cindy. Their support, patience, and mentorship have been invaluable for me, and I'm really grateful for how much they've helped me grow as a scientist. Thank you.
All right, thank you, Yun. Next, we have Cindy van Belthoven, who is also from the Allen Institute, where she is an associate investigator on the informatics and data science team.
Okay, now I'm unmuted. Thank you, Rachel, for the introduction. So we took a deep dive into the transcriptomic and spatial organization.
Hi, Cindy, are you with us?
Yes, I think. I don't know what happened.
Okay, we see your slides now.
Okay, perfect. Okay, so we took a deep dive into the transcriptomic and spatial organization of GABAergic neurons in the mouse telencephalon. In 2023, we released a whole mouse brain cell type taxonomy. This is a hierarchical cell type taxonomy that was created from single cell RNA-seq data and a spatial transcriptomic Murphy's data set of covering the entire adult mouse brain. In the accompanying manuscript, we described the landscape of all these cell types and the relationships between these types at the subclass level, which is a relatively high level. So for this specific paper, we kind of isolated the subpallium GABAergic neighborhood and took a closer look at the organizing principles within this specific group of telencephalic GABAergic neurons. So this neighborhood consists of 615,000 cells, and we broke that down into 7 classes, 51 subclasses, 285 supertypes, and a little over 1,000 different clusters. So this hierarchy that you see here is the hierarchy at the supertype level. And below it, you see a breakdown of the region that the cells were dissected from. And what I hope you can see is that some of these classes are very regional specific. So for example, the lateral septum class contains mostly cells coming from lateral septum, and the same for olfactory. But then some of these other classes contain cells coming from different regions. We use a lot of different visualizations to take a look at the relationships between types and how they're organized. So on the left, you see UMAP at the subclass level, so the intermediate level in the hierarchy, and a constellation plot of those subclasses. In this constellation plot, which is based on the UMAP, each centroid position is the centroid of the subclass in the UMAP space, and the size of the node tells you something about the size of the number of cells that are included in the cluster. The edges between the nodes show how many of the neighbors of each of the cells are shared between the 2 different nodes that are connected. And then we have the spatial transcriptomic data that helps us really put all these transcriptomic types into a spatial location. So in this case, the Murphy sections that are showing the class labels, so very broad, high-level labels. But you can see that these classes are very tightly distributed within the brain. This is a similar constellation as shown in the previous slide, but now really showing it at the supertype level. And there's a lot that you can glean from a constellation plot like this, as well as from the different UMAPs and the spatial transcriptomic data. In this specific constellation plot, the kind of the left to right transition in the constellation is related to kind of a dorsal to ventral positioning within the brain. So in this outer ring, you have like the types that are located in cortex and olfactory. And these types tend to be somewhat bigger, tend to be more transcriptomically distinct from each other. Then in the middle, you have these types, these classes that are populating the cerebral nuclei, which includes the striatum. The LGE-derived GABAergic types tend to be somewhat bigger and tend to be more distinct, whereas the MGE-derived CNU types tend to be more transcriptomically similar. And then we have like the most ventral classes, the lateral septum, and the anterior hypothalamus, where we have a lot of smaller supertypes that are highly similar to each other. In the manuscript, we describe a variety of kind of discrete and continuous gene expression gradients that are running through all these different types, and some of which are associated to spatial dimensions as well. These spatial and molecular gradients, they don't only organize the cell type diversity within the adult mouse brain, but also suggest kind of deeper rules of specification of these types, rules that we can potentially uncover by looking at the transcription factor profiles. So in this study, we identified a large set of transcription factors that are specifically marking classes and subclasses and supertypes that we have defined in our transcriptomic data set. And several of these are key factors that play a role in early developmental processes. And there are some examples below the heat map where the first images are from the ISH atlas showing the expression of specific genes at E13.5, and then the expression of that same gene in our adult merfish data set, where a lot of these patterns are very highly localized in the embryonic period. A lot of these, the expression patterns tend to be more widespread in the adult brain. So to take a closer look at where these cells are coming from, so in the mouse, most of the GABAergic cells are derived from 5 different progenitor domains within the embryonic subthelium. From there, they proliferate and migrate and ultimately differentiate and incorporate into the developing circuit. And so to trace back where these cells are coming from, we generated an additional data set covering 9 different time points from E11.5 to P14. And we integrated this with external published data sets that are mostly covering the embryonic period from E8 to roughly P3. So in total, we have 614,000 cells coming from the developing cells that we can integrate with the adult data set. So this is what the integration then looks like. These are UMaps. The first one is colored by age. With dark red is the P56. And then it's more like the more you go to the middle, the younger the cells are getting. The middle is the classes that we have assigned to each of these cells. And then on the right-hand side is the adult subclass that has been assigned in the adult mouse brain taxonomy. So the labels were transferred in a method similar to what Yuen just described, where we transferred labels from P56 to P14 and from P14 to P12 and so on. And as expected, a lot of the transcription factors that we've identified previously, we can trace these back to specific progenitor domains early on. So when we take into account the spatial location that we've identified in our adult merfish data set, this really indicates that most of the GABAergic neurons really have migrated over long distances to reach their final destination. But these long kind of migratory journeys are really just one step in the process. As these cells are dispersing from their progenitor zones, each lineage will undergo further diversification, which gives rise to kind of the rich repertoire of cell types that we observe in the adult taxonomy. This diversification, however, happens at very different rates. So in the dorsal classes, these cortical classes coming from CGE and MGE, the neurons diversify extensively, but mostly later on, so after birth. Whereas in the more ventral classes, the lateral septum and the anterior hypothalamus classes, there appears to be kind of a burst of diversification in the embryonic period, where most of the types tend to kind of arise all at the same time. This pace of diversification mirrors some of the cluster properties that we've seen in the adult types. So the ventral classes show a rapid kind of embryonic burst of diversification, and you yield a lot of small, tightly related types, whereas the dorsal classes continue their diversification postnatally, and they give rise to the larger, more distinct types. In summary, like I just mentioned, the dorsal forebrain structures have larger, more distinct clusters, whereas ventral forebrain structures have like smaller, more similar clusters. We've identified various spatial dimensions of discrete and continuous variations in the cell types that we've identified. Long distance migration and dispersion happens to kind of nearly all of the classes of the telencephalic GABAergic neurons. And there are distinct developmental timelines for the diversification of these cells. And with that, I would like to thank everyone who was involved in getting all this work done. Thank you.
Thank you, Cindy. Next, we'll move on to the next talk by Emily Corrigan, who is a PhD student in the Developmental and Stem Cell Biology program at UCSF.
Can you see my screen?
Yep, we can see your screen.
Okay, great. Thank you for that introduction. And hello, everyone. I am Emily Corrigan. I am a fifth-year grad student in the Pollen Lab at UCSF, and I'm really excited to share our recent study titled Conservation and Alteration of Mammalian Striatal Interneurons. So broadly, the theme of our paper is how evolution can modify the brain on a cellular level. And so first, evolution can modify brain structures by simply increasing or decreasing cell numbers. Cells can also move to new locations. There can be molecular changes in conserved cell types. And then finally, evolution can simply invent a new cell type in one species that others do not have. In fact, a few years ago, a group was performing single-cell RNA sequencing on the human and marmoset striatum, and they observed a transcriptionally distinct interneuron type that had not yet been described, and it expressed the neuropeptide TAC3, thus giving it its name, the TAC3 interneuron, and it made up about 30% of the striatal interneurons in these species. And then when they surveyed the mouse striatum, they didn't observe this population. And finally, when they surveyed the ferret striatum, they also didn't observe this population. And so due to its absence in mouse and ferret, this cell type appeared to be a primate-specific population. Luckily, its transcriptome gave us some insight into where this population may be coming from during development due to its expression of the transcription factor NKX2.1. So NKX2.1, along with LHX6, is the signature marker of the medial ganglionic eminence, or MGE, pictured here in blue, in both the developing mouse and developing macaque brain. The MGE is a highly conserved birthplace for the majority of both cortical and striatal inhibitory interneurons. And so following the final cell division within the MGE, the initial class of an interneuron is specified. Then these newborn neurons migrate to their final destinations, and it's during this migration and after that they undergo post-mitotic fate refinement to form their terminal class. And so the TAC3 interneuron itself is the terminal class, but our lab is really interested in development. And so for us, a question that we had is, what is the initial class that gives rise to the TAC3 interneuron? And we addressed this in some previous work where we examined single-cell sequencing of mouse and primate interneurons during development and then mapped them to terminal classes. And so what I'm showing here is really just a fraction of that work, but it was shown that the majority of both initial and terminal classes of inhibitory interneurons are conserved across mammals, with the exception of both the initial and terminal class of the TAC3 interneuron being primate-specific. But there's been some recent work in other systems that has revealed that populations that we thought were primate-specific actually have orthologs in other species. And so seeing work like that really motivated us to examine the phylogenetic extent of this initial class in other mammals. Which brings me to our new study, where we now survey approximately 160 million years of evolutionary divergence, stretching from primates to marsupials. We perform single-cell RNA sequencing on developing striatum and cortex samples from 7 additional species to examine inhibitory neuron initial class diversity across a total of 8 species. And so after isolating inhibitory neuron populations in each of these species, we then embedded them in a shared latent space. And here I'm showing that integrated UMAP colored by each individual species contribution. And because we were able to integrate all of these 8 species, we were then able to label populations based on conserved and well-characterized markers of initial classes of inhibitory neurons. And we saw that cells across the 8 species group by cell type rather than by species, and that all initial classes were conserved and highly correlated across all species sampled. But what really grabbed our attention, however, were these MGE-derived CRAB-P1 expressing classes. We saw, surprisingly, that again, all 8 species contributed to both CRAB-P1 expressing clusters, including the TAC3 initial class cluster, which was previously thought to be primate-specific. And when we examined the gene expression of the TAC3 initial class in each species, we were able to conclude that the TAC3 initial class is conserved across placental mammals and exhibits both marker conservation and marker turnover. And so what I mean by that is we saw this initial class is really defined by a core set of markers, including MGE lineage-based markers like NKX2.1 and LHX6, as well as other markers like ATV1, ChRNA3, acetylcholine receptors, and Zik markers. But then when we examined individual species more closely, we were also able to see species and clade-specific gene expression. And so, for instance, we saw that marsupials actually lacked cellular retinoic acid binding protein, or CRAB-P1, while macaques seemed to be enriched for this BMP antagonist SIR1. But what was most striking about this is that when we looked at the mouse, we saw the loss of TAC3 in this initial class alongside the gain of TH. So this suggested that the TH interneuron, which was previously thought to be an entirely different cell type, is actually homologous to the TAC3 interneuron. And to further examine the possible homology of the TAC3 and TH interneuron types, we performed integrations of adult mouse and primate striatal inhibitory neuron datasets. And so first, we saw the integration of major subtypes between these primate datasets, including a large TAC3 cluster here, a subset of which expresses LHX8. And then when we looked at the integration of the mouse, we found that the TH interneurons integrated the most with the TAC3 type in primate. We further validated this homology using SAMMAP, where we observed high homology scores between the mouse TH interneurons and primate TAC3. We performed additional tests, including gene module tests and Pearson correlations, which further supported these results, but I unfortunately don't have time to show them today. We also independently examined the gene expression of the mouse striatal inhibitory interneurons, and within the TH interneuron cluster, we observed a very small population of interneurons that express residual TAC2. So TAC2 is the ortholog of TAC3, so same gene, just a different name. And then using the Allen Institute's MERScope data, we found that this TAC2 expressing TH population was localized primarily within the ventromedial striatum. And so taken together, we show that this CRAB-P1 TAC3 initial class is conserved across placental mammals, with this conserved initial class producing TAC3 interneurons in most mammals and TH interneurons in rodents, a small subset of which retains that residual TAC2, and it's restricted to the ventromedial striatum. And broadly, we think that this gives us some more insight into how evolution modifies the brain on the mammalian timescale. We believe that it tends to favor teaching old cells new tricks rather than inventing entirely novel cell types early in development, with TAC3 interneuron evolution actually encompassing all of these mechanisms. I didn't have time to go into it today, but we do see a lower proportion of TH neurons compared to TAC3 neurons in the primate. We also observe this initial class migrating to the cortex in the pig and ferret, which is not observed in the primate or mouse. And finally, of course, we see modifications of its gene expression throughout evolution. And with that, I want to thank you for your attention, as well as my lab and all of our amazing collaborators.
Thank you, Emily. Next, we have Marilyn Stiert, also from UCSF, where she is a PhD student in the Biomedical Sciences graduate program.
Hi, can everyone see my screen?
Yep, we can see your screen and your cursor.
All right. So today I'm excited to share a lineage resolved atlas of the developing human cortex that I've been working on alongside a now graduated PhD student, Matt Keefe, in the lab of Tom Nowakowski. And it's an honor to be here sharing this work. Okay, so it takes no introduction to this crowd to say that there is massive cell type diversity in the adult mammalian brain. And of course, since we're interested in development, we really want to know, how does it get here? How does it arise from the relative simplicity that exists during development? And progenitors give rise to, of course, more progenitors, but also these more distinct cell classes over time sequentially. So lineage tracing, we think, is a really powerful tool that sort of focuses in on the arrows on this map and allows us to look at the connecting individual progenitors to their cell type output. We use lineage tracing to generate an atlas of human cortical development, sampling across a couple of developmental stages, and also 2 different germinal zones to measure the lineages from 2 different progenitor types, truncated and outer radial glia. So I'll kind of come back to that and start with the developmental stages. But first, how did we do this? We used a tool previously developed in the Nowakowski lab called Sticker, which has these long, complex barcodes and can be packaged into a lentivirus, which we could use to infect the tissue. And we actually infected the tissue with 2 different pools of this lineage tracing tool so that we could read out the differences between these 2 progenitor outputs. And we cultured the cells for 10 to 12 days to allow the lineages to take shape. Afterwards, we sorted out just the lineage trace cells, which expressed GFP, and could use single-cell transcriptomics to read out the cell types that we had traced from and the clonal relations between them. So we collected dorsal progenitors and their lineages, sort of got the expected cell types plus some interesting ones, and we had about 8 biological samples, and we collected over 6,000 multicellular clones. So to sort of get us started analyzing this, we aggregated it all together and tried to get a picture of the global lineage dynamics in a graph that looks like this. So we could see that, for example, there are a lot of radial glia shown here in pink, and their clonal relations extend across many cell types, whereas some other cell types were more likely to be in shared clones with only themselves. We saw that there was a shift in clonal relations across mid-gestation. So in the younger samples, we saw lots of these excitatory neurons, and in older samples, we saw an uptick in generation of oligodendrocyte precursor cells, shown in yellow, as well as these locally born interneurons and their progenitor types, shown in blue at the top. And to dig a little bit further into this cell type, because it seemed really interesting to us, we validated the identity of these cells and found that they have a distinct identity, which could not be found in mouse samples. And they specifically co-express secretagoguin along with PAX6. And we could stain for this in uncultured tissue and find them in the cortical plate, especially in the upper layers, suggesting that these cells were actually being born and migrating out to their final destination. So in the beginning, I introduced that we used 2 different pools of this lineage tracer to look at the difference in output from outer radial glia and truncated radial glia using their spatial separation. And we did this because very little is known about the output or the role of truncated radial glia, since their transcriptomic and morphological description by work that was done in the Kriegstein lab about 10 years ago now. And so we learned a couple of very interesting things. We learned that, first of all, truncated radial glia are highly self-renewing. This shows that, so we looked specifically at clones where we actually collected something we thought was a truncated radial glia based on its transcriptomic identity. And we found that they were often in shared clones with other radial glia. So these could be more truncated radial glia, or they could also be outer radial glia. The next most likely thing that they were to be in a shared clone with was excitatory IPCs, and we also saw some evidence of them being in the same clone as excitatory neurons. So this suggests that they're actually quite highly neurogenic, which was a bit surprising. We really expected them to be more gliogenic, if anything. So we wanted to dig into this a bit further and validate. and see if this was happening also at late stages. And we looked specifically at the late stages. This is the UMAP with the teal representing cells that came from the VZ and coral representing cells that came from the outer subventricular zone. And we could see that both of these cellular compartments, even at these late stages, were making excitatory neurons. We validated this, especially in the VZ, by staining for NeuroD2, which is just a marker of excitatory neurons. And we found some cells expressing this marker in the ventricular zone, even as late as gestational week 24. So really sort of an unexpected finding. And we wanted to know, what is the layer identity of these late-born excitatory neurons? We know that the cortex is canonically born from the inside out. And so we wanted to see if this was true. And much to our surprise, we found that they were actually deep layer excitatory neurons. So the evidence for this was that so these are stains from the outer subventricular zone and the VZ, ISVZ region with a deep and upper layer marker, TBR1 and SAPB2 respectively. And you can see at early stages, there's just a lot of neurogenesis, both upper and deep layer neurogenesis. At later stages, we saw a marked decrease in SAPB2 positive cells in both of these compartments, which was surprising in and of itself. But we also saw continued expression of TBR1 positive cells in the ventricular zone compartment. This suggested that deep layer excitatory neurons were still being born in the VZ, even at these late stages. And we validated this finding using BRDU birth dating, where we could label just the VZ with BRDU and track the cells that were born from that compartment in late stage tissue samples. And we looked at the subplate and deep layers of the cortical plate and found BRDU positive cells that express TBR1 but not SAPB2, suggesting that they really are deep layer neurons, not like upper layer neurons that could be migrating through or anything. This was really surprising, but it did sort of explain sort of a long-standing question in the field. We know that the human subplate expands well after subplates were thought to be born, which really are thought to be the earliest born cells of, or excitatory neurons of the developing cortex. And so potentially the presence of truncated radial glia and their contribution of subplate neurons late is how the human subplate keeps expanding for so long. So to conclude, sort of the major findings from the study were that dorsally derived GABAergic neurons possess a unique transcriptomic identity. They express secretagoguin and PAX6 together. The truncated radial glia give rise to glutamatergic neurons, and surprisingly, these are late-born glutamatergic neurons that take on a deep layer identity. Really, the summary of this, I think, and sort of a bigger lesson that we learned from this study was that there are interesting and unique lineage dynamics that happen, especially in the later stages of mid-gestation. And these were only resolvable because we had the lineage tracing tool at our disposal. We hope that these data will be useful to the community. They're available on the UCSC cell browser. Hopefully a link will be shared to that soon. And I want to thank especially Matt and Tom for their contributions to the study. It was really a joint effort between Matt and I, and Tom's mentorship is amazing. And of course, I'd like to thank the funding and everyone else that contributed to the study. And thank you for your time.
Thank you, Marilyn. Next up, we have Harris Kaplan, who is a postdoctoral fellow at Harvard University in the Department of Molecular and Cellular Biology.
Okay, thanks. Hi, everyone. I'm very excited to present our work alongside all these wonderful studies today. So I want to start with the whole mouse brain transcriptomic atlas, which, as Cindy described earlier, was published a couple of years ago by a large BICCN-funded consortium. So using single-cell sequencing, they cataloged over 5,000 transcriptomically defined cell types across the entire mouse brain. And importantly, they used spatial transcriptomic approaches such as MERFISH to show where those cell types are localized in different brain regions. And that's summarized in this plot here. So each column here is an individual cell type, and each row is a kind of major brain region. And so you can see which cell types fall into which regions. And one of the most intriguing findings here is that the vast majority of neuronal types are actually subcortical. And one way to think about this is that in Cortex, there are fewer cell types that together form a kind of general purpose circuit, which is the six-layered circuit we're all familiar with. And this one circuit is capable of very kind of flexible computations. Whereas a subcortical region like the hypothalamus has many diverse cell types, about 900 or so. And these cell types are thought to have a much more dedicated control over specific behaviors. And I think this is interesting, especially in the context of development, because I think we know much more about the development of this small sliver of cortical cell types and relatively little about how the vast majority of neuronal cell types develop. So for example, in the preoptic area, which is part of the hypothalamus, at the bottom of the mouse's brain here, this area is involved in different homeostatic functions like thirst, sleep, and thermoregulation, as well as different social behaviors like parenting, aggression, and mating. And many labs have shown that there are transcriptionally defined cell types within the POA that are dedicated to each of these functions. So just like I described in the previous slide, this subcortical region, the POA, has individual cell types that are dedicated to specific functions, but not others. And so we know a lot about the POA in terms of the functions of individual cell types, but all of this work comes from studies in adults. At the beginning of this study, these cell types had really not been described at any earlier age. And so my goal has been to address this question: how do POA cell types develop? And so I established a single nucleus RNA sequencing atlas of the mouse preoptic area at the 8 ages shown here, from the end of neurogenesis at E16 all the way to adult at P65. So for each of these 8 ages, I dissect out the POA, extract nuclei, and perform sequencing. And so overall, I profiled over 200,000 nuclei across the 8 different ages. This allowed me to define about 150 different neuronal cell types shown by the different colors here, 64 excitatory and 83 inhibitory subtypes. Importantly, these cell types were identified by mapping them to previously published MERFISH atlas of the POA. And because that atlas has spatial and functional information, I can kind of inherit that information into my data set. And so I'll tell you about 2 key questions we asked using this data set. The first is, how does the POA generate this really staggering diversity of about 150 neuronal cell types? And so here you see the same UMAP colored by cell type or by age. And what we see is that each cell type consists of cells from all 8 ages in this beautiful gradient pattern, which is kind of repeated throughout this plot. And so what really surprised us when we looked at these data was that even at E16, these cells are already grouping with just a single adult cell type in UMAP space, which suggests that they're already diversified really early in life. And so to quantify this, we turned to label transfer. And what we found is that indeed each E16 cell type maps in essentially a one-to-one fashion onto the adult cell types. And so this tells us that the full complement of about 150 cell types is already present at this really early age of E16. And so this prompted us to ask when this diversity first emerges. So we decided to sequence at E14. And we chose E14 because this is roughly the midpoint of neurogenesis in the POA. So at this time, about half of the cells are still dividing progenitors, and the other half are newborn neurons, which were born in the previous 1 or 2 days. And so we could ask how diversity looks like in each of these populations. And so first, for the neurons, we find that, again, they're largely diversified already at E14. We can map them onto the adult cell types in a one-to-one fashion, whereas the progenitor clusters largely don't map. And so this tells us that the E14 progenitors really lack any mature regional or cell type identity, which means that these neurons diversify really rapidly upon neurogenesis in just 1 or 2 days. And so to summarize this first part, we find that these POA neurons diversify rapidly and very early in life. And one idea is that this makes sense because the POA is important for functions that might be essential really early in life, like thirst or sleep, as opposed to maybe cortex, where cell types emerge more gradually and maybe don't need to be diversified until cognitive functions emerge later in life. And so next we asked whether POA development is sensitive to environmental inputs. Decades of research has shown that sensory circuits, for sensory circuits, there are critical periods in development where, for example, visual experience affects the development of visual cortex. And we know that deprivation of early life social experience can lead to long-lasting deficits in social behavior, for example, from decades-old social isolation rearing studies in primates. And yet we don't know how social inputs affect the development of circuits involved in social behavior. And so to address this, we decided not to use social isolation, but instead took a more targeted approach by sequencing in different mutant mouse lines that are impaired in different sensory modalities like olfaction, pheromone sensation, touch, or thermosensation. And so for each of these mutants, we sequenced the POA in mutants and littermate controls, focusing on 3 key ages when we thought social input might be especially important. And so for each mutant, we can ask how each POA cell type is affected. And to do so, we used a distance metric in PCA space to simply ask if the mutant and control cells kind of cluster separately. And what we find is that for the mutants impaired in touch sensation, no cell type is significantly affected across these replicates. For mutants impaired in thermosensation, a couple of cell types show small effects, but nothing really major stands out. Similar for the mutants involved in olfaction, whereas for mutants involved in pheromone sensation, we found that many cell types are affected, and that the effects are actually really significant, really large. And so what is this mutant TRPC2? So TRPC2 is expressed in the vomeronasal organ, or the VNO, which is an accessory olfactory organ that's present in most mammals and which is specialized for pheromone detection, so for detecting these chemical signals for conspecific sex, age, or state. And pheromone information from sensory neurons in the VNO reaches the POA down here in just 3 synapses. And so TRPC2 is a gene that is expressed almost exclusively in the VNO sensory neurons, and it's required for sensory transduction in those neurons. So in TRPC2 mutants, pheromonal information does not reach the POA. And so what we find is that in these mutants, many cell types are affected, but only actually at specific ages. So not at birth or in adults, but only at these kind of intermediate ages of P10 and P18. And so to understand this better, we quantified the gene expression maturity of these neurons using a pseudotime analysis. And so you can see the control cell types show increasing maturity in gene expression across age, whereas the TRPC2 knockout cell types are less mature, especially at these intermediate ages of P10 and P18. And so from these data, we conclude that TRPC2 mutants show a developmental delay in the maturation of POA cell types. And so what we think is happening is that at these intermediate ages of around P10, pheromonal information does not reach the POA, and this leads to a delay in POA cell type maturation. And due to previous work and other analyses, we think that this might lead to long-lasting effects on POA cell type function and adult social behavior. And so in conclusion, we find that POA cell types show a very striking pattern of early diversification, as well as a sensitivity to early life environmental inputs, especially pheromonal input. And so with that, I want to thank all the co-authors, the Duoloc Lab, and collaborators, and especially the BICAN program. Thanks so much for listening.
Thank you, Harris, for that presentation. Next, we have Camille Mannens, a postdoctoral researcher at the Flemish Institute for Biotechnology.
All right, thanks, Rachel. Yes, so this is a project that I conducted during my PhD in the Stan Linnerson lab, where we looked at chromatin accessibility during the first trimester of human neurodevelopment. So an ongoing project in the lab over the past 10-15 years has been the aim to create a comprehensive atlas of the human brain in collaboration also with the Allen Brain Institute, which has resulted eventually into landmark papers characterizing the cell types both for the adult brain as well as the first trimester in development. But while working on the developmental part, we started to realize that for some of the kind of key questions that we wanted to ask about cell-fate transitions, we needed to look also beyond single-cell RNA-seq, which we had worked with primarily before, and look into gene regulation, primarily in the form of chromatin accessibility. So this is exactly what we did. We collaborated with the clinic at the Karolinska Hospital, as well as the Human Developmental Biological Resource, and Roger Barker at the University of Cambridge. And we collected 26 embryos, staged between 5 and 14 post-conceptional weeks, for which we conducted either single-cell ATAC sequencing or single-cell multi-omi sequencing. The multi-omi we strategically placed along the timeline to try and capture all the different time points so that we could impute then gene expression across the rest of the data set. And all these samples were dissected into the different prosomeric regions, so we could compare between regions and identify specific cell types for each of them. So this resulted in a data set of about 500,000 cells, where we could compare gene expression with accessibility of the gene regulatory elements driving the expression. And we could identify, for instance, genes that were primed but not yet expressed, and vice versa. But perhaps more interesting for the rest of the talk is that we also looked extensively at gene regulatory elements and what defines their specificity in relation to cell types. In most cases, this means that we're looking at transcription factor binding motifs. And kind of nicely illustrated also before by Cindy and Emily in the ganglionic eminences, where many of the interneurons are born. There's an early differentiation between the two regions of the medial and the lateral ganglionic eminence. And to kind of illustrate why these multi-omic approaches can be quite useful, we identified that the key regulators for those two regions were both homeodomains, homeodomain transcription factors, which you cannot find the difference just by the binding motif, but with the RNA data on top of this, we could identify LHX6 and DLX2 as the two kind of central players in that field. So just to kind of illustrate the approach here. Next, I'll show a few vignettes from the paper, interesting things that we did. One of the things that we've been interested in for a long time is the switch from radioglia to glioblasts and the timing there. So when you look at the different parts of the brain, this switch occurs at different time points. So radioglia in the telencephalon, for instance, exists throughout a very long time in development, whereas in the hindbrain, they start to switch to a glioblast state quite early, which means that they stop producing neurons and only produce glial cells like OPCs and astrocytes. And we wanted to look at some of the regulators that cause this switch. So we did differential abundance testing related to the time points and identified NFI binding factors as important here for that switch. But it was difficult to see if this is a timing effect or if this was primarily due to the switch from radioglia to glioblasts. So we decided to also look at the other cell types that we had in the data set and compare the earlier to the later versions there. So for instance, we would take the neurons and the neuroblasts, split them between the earlier cells, so the ones from before 10 post-conceptional weeks and those after, and then identify the transcription factors there that were different. And we found that the NFI transcription factors, in fact, were enriched in most of these later cell types. So it seems to be an important timing regulator rather than necessarily just for glioblast transitions. We also found, on top of that, that many of the later cell types tended to have more accessible regions than the earlier cell types, kind of indicating a more specific regulatory programs being active there. Then we wanted to dive in a little bit more in trying to characterize the gene regulatory elements and what kind of networks of co-accessibility that they form. So we did topic modeling on all of these regions based on their activity in different cell types, and I identified about 175 different topics. So here on the left, the TISNE, these are genomic elements, not cells. And we found that quite a lot of them had very distinct programs that they could be labeled as. Many of these programs are related primarily to cell types, but some of them also showed broader functions. So for instance, we could identify that many of the TSS sites form a single cluster as well. But most interesting to us was that we found that there were two topics for CTCF binding sites. So these, of course, are important in the formation of cohesion loops and transactivating domains. And that these topics were specific either to the glial state or to the neuronal state. So that means that from the intact data, we could see the formation of this new layout of the chromatin in the sense of the transactivating domains, which was quite cool. Then finally, we also trained a convolutional neural network, so a sequence to function model, with the aim of taking as inputs different gene regulatory elements and trying to predict what kind of cell type they would be active in. And then you can use these models to also backpropagate through them to try and understand what parts of the input sequence were important for the prediction. So we did this using PyTorch. If you're interested in trying anything like this, there's also now some packages that do a lot of the heavy lifting for you. But we use this model to then identify the sets of transcription factor binding motifs that the model picks up as being the most important ones for identifying specific neuronal cell types. For that, then to conclude, this larger data set allowed us to investigate some gene regulatory processes in more detail. We could use the data also to do enhancer syntax learning. And not shown here, but if you're interested, there's also in the paper some more figures on the temporal delays that we see in enhancer activation and how we can explain those based on the presence of specific transcription factors, and integration with GWAS data to try and prioritize vulnerable cell types in psychiatric disorders. With that, I want to thank everybody in the lab, all the collaborators as well, the funding agencies, and the donors who make this work possible. Thank you.
Thank you. All right, now we'll move on to our final speaker of the day, Li Wang from Stanford University, an assistant professor in the biology department. And this is our last presentation of the webinar, and we will have some time for some brief question and answer at the end. So please continue to enter your questions in the Q&A box.
Hi, everyone. This is work done in Dr. Arnold Christen's lab and at UCSF, and I was a postdoctoral fellow. And today I wanted to share with you this paper we just published on defining the molecular and cellular dynamics of the developing human neocortex. The neocortex in human is largely expanded compared with the other species, and the neocortex contains about 16 billion neurons in the human, and it is important for planning, reasoning, language, and many other behaviors that humans excel at. Therefore, we really need to understand the neocortex, in order to understand the uniqueness of the human brain. And many of the other speakers have already mentioned the developmental state, how the neocortex is developing in human. There are certainly many different cell types, including progenitor cells like radioglial cells, neurons, and both inhibitory and excitatory neurons, as well as glial cells such as OPC and astrocytes. So this is a really a complex structure, therefore, and also be regulated by both intrinsic factors such as gene regulatory networks, as well as extrinsic factors such as cell-cell interactions. Therefore, to truly understand how the whole process is being regulated, we need to understand its single-cell resolution across modalities. To do that, we have collected human neocortical brain samples from the first trimester and all the way to adolescent stages. And for the earlier stage samples, we focused on the whole neocortex, but for later stages, we focused on two major regions. One is the prefrontal cortex, the other one is the primary visual cortex, to highlight regional differences. We collected the samples and performed nucleic preparation, 10x multiome sequencing to get the epigenome and transcriptome information from the same nuclei. In the resulting data sets, we identified in total 33 major cell types in the developing human neocortex, including different subtypes of the neural stem cell, radioglial cells, excitatory neurons, inhibitory neurons, and glial cells, including astrocytes, OPC, as well as oligodendrocytes. We also went further to understand the spatial context of these individual cell types. So we performed morphology analysis by designing a 300 gene panel based on the single nucleus multiome data. Here I'm showing you 6 of those 300 genes, and these are known to be layer-specific markers. They're already demonstrating layer-specific patterns in this second trimester human neocortex. And based on these 300 gene markers, we can further define the cell types, including different excitatory neuron subtypes in the cortical plate, as well as neural stem cell subtypes in the germinal zone. And then based on the local composition of cell types, we can define domains, including cortical layers, as well as germinal zone layers. Perform such spatial transcriptome analysis across 3 different major developmental stages, from second trimester to infancy, and also across 2 major locations from PFC to V1. Wanted to mention that these cell types are defined based on the markers we derived from the single nucleus motium. Therefore, we have one-to-one correspondence of these cell types to the single nucleus motium data, allowing very streamlined integration. We identified in total 10 different domains within the developing human neocortex, again, across all the cortical layers, as well as the subcortical regions, including the germinal zone and white matter. So based on these rich data set, we have gained multiple biological insights into human neocortex development at both molecular and cellular levels. One of the interesting things we decided to pursue is a gene regulatory network that regulates fate decisions of excitatory neurogenesis. To do that, we have identified these irregulons, which consist of the transcription factor, its target enhancers, and target gene activities across individual cell types. You can see many of them are displaying cell type-specific activities collectively. We also integrate that information with a trajectory analysis and identified bifurcation point where each of the neurons have to decide which trajectory to follow to become a terminally differentiated neuronal subtypes. At each of these bifurcation points, you can identify key transcription factors that are differentially have differential activity across the 2 emerging branches, and suggesting that they are key regulators of the fate decision of those neurons. We also integrate the single nucleus motium data with our spatial transcriptomic data to identify cell-cell interactions. And in this case, we're employing this node-centric expression model analysis, node-centric expression model analysis by understanding how a particular cell type as a sender cell will affect the transcriptome of another cell type nearby as a receiver cell. You can see that many excitatory neurons are indeed affecting the transcriptome of nearby excitatory neurons, but we also find very interestingly that inhibitory neurons are able to regulate the nearby excitatory neuron transcriptome even at this very early stage. So we think that probably besides synaptic connections, there may be something that is something such as secreted factors that are regulating the nearby excitatory neurons by inhibitory neurons. And we find that one particular inhibitory neural subtype, somatostatin positive inhibitory neurons, indeed are predicted to be able to regulate or impact the nearby excitatory neurons through the somatostatin signaling. And further validation experiments have demonstrated that activating the somatostatin signaling actually delays the maturation of excitatory neurons across different neuronal subtypes. We also try to understand the lineage potential of individual progenitor cells in the human neocortex by performing prospective lineage tracing. To do that, we performed tissue dissection, fact sorting based on cell type specific markers for different neuroprogenitor cells at this developing human neocortex between gestation week 20 to 24 to understand which progenitor cells are actually important for the switch from neurogenesis to gliogenesis. And we focus our analysis on 3 of them, the truncated radioglia and alpha radioglia, and this so-called IPC glia population based on their cell surface markers. By tracing their differentiation using single-cell RNA sequencing, we find that this IPC glia population actually gives rise to 3 different lineages, astrocytes, OPC, as well as interneurons, but not excitatory neurons, suggesting that they're tripotent progenitor cells, and therefore we have named this population as tripotential intermediate progenitor cell, or TRIPC. On the other hand, you can see that the TRIPC population can actually be produced by both the truncated radioglia and alpha radioglia in 2D culture, and these cells will further give rise to astrocyte, OPC, and inhibitory neurons later during the culture. We also demonstrate the tripotency in vivo using transplantation assays. And interestingly, this population seems to have some transcriptomic similarity to cancer cells in glioblastoma, suggesting some of the developmental mechanisms may be hijacking tumor as well. Finally, we also wanted to understand that if there's any enrichment of particular neurodevelopmental or psychiatric disorders in particular cell types at specific developmental stages using our data set. So in this case, we integrate the GWAS data with the epigenome data to understand how the common variants are enriched, are associated with different disorders, are enriched in the open chromatin regions of different neuronal subtypes. Here I'm showing you two examples. One is autism spectrum disorder, the other one is bipolar disorder. As you can see, they indeed displayed different enrichment patterns across all neuronal subtypes, with autism spectrum disorders primarily risk are primarily enriched in immature excited neurons you find in the second trimester. But for bipolar disorders, those are mostly in the later stage neurons as well as inhibitory neurons. We can also group these risk scores based on developmental stages from the first trimester all the way to adolescent stages, and consistent with previous analysis using rare variants in autism spectrum disorder. We find that the highest risk for the disorder has been concentrated in the second trimester from the common variants. But for bipolar disorders, you can see that the highest risks come actually from infancy stage, suggesting disease-specific risk factors that affect the human cortical development. All of these data are available from this interactive browser, and hopefully you can also take advantage of this data to obtain new biological insights beyond what we have demonstrated. With that, I'd like to thank Dr. Arnold Krigstein for his fantastic mentorship, as well as collaborators, Dr. Jingjing Li and Dr. Chen Wang, who have contributed substantially for this project, and many collaborators at UCSF and funding sources from the BRAIN Initiative, as well as other sources. I'm happy to answer questions as well. Thank you for your attention.
All right, we have just a few minutes left for questions. So please, if you have any questions, submit those in the chat box in the Q&A function. And one question that we can start with is, what are some ways that others can use some of these larger data sets to influence their own work? Or how can they use some of these data sets in their own labs?
I'm happy to take this one if no one else is speaking up. I know that there's a lot of resources and that there's a lot through the BRAIN Initiative as well as through the Allen Institute, and these include places to download data. You can get them from NeMO, which is also a repository, and there's many interactive pieces. I also want to highlight that others have also engaged with the UC Santa Cruz cell browser, which is also an interactive tool that requires no informatic expertise to engage with it, but then also if you want to download the underlying objects. So hopefully each of the papers and the BRAIN Initiative and BICAN websites have links to many of these tools that allow for both interaction as well as download and engagement with the underlying data.
The links to NeMO are in the first or second question in the question box too, but I think Aparna's right, they're in the resources as well, the papers.
And this webinar is being recorded. It'll be up on the BICAN YouTube channel afterwards. So if you've missed some of the URLs that were linked in the slides, don't worry, this will be posted online later so you can review and pause at those screens and get some of those URLs.
I'd like to make a maybe perhaps final comment. I think there are a lot of questions about the resources, with all these different great data sets, how they can be accessible. Right now, many of the data are being made available in the individual labs or Allen Institute, that can be found in the individual papers. But I think, in BICAN collectively, we should have some kind of a central page to list all the availabilities of the different resources, data sets, and things like that. This is something that we should work on going forward. Already in the plan, but it will be realized in the coming year.
All right, I just saw in the chat that we had a last question was just answered by Li Wang. Which disease association analysis did you use? And the response was, we apply a method called scavenge, for which details can be found in the link there. All right, so thank you all of our speakers for a great webinar today and all these wonderful talks. And again, this will be up on the YouTube channel afterwards if you'd like to rewatch or revisit some of these slides. And thank you everyone for attending today's webinar.
Thanks, everyone.